My name is Vinny Therasala, and I'm here today to talk to you about FlowPoint Biotech. We are unlocking a new frontier and changing what's possible in the world of cell engineering. Like it or not, cell and genetic modification therapeutics, where we can reprogram the body's own immune cells, is the future of medicine. It's already poised to help millions that are in need of better and safer cancer therapies fight their disease for good. There are over 40 approved products already, running the gamut from CAR T cells, pluripotent stem cells, and more that are actively in use. And these therapies are only going to become more and more prevalent over time. However, broad access is going to be hamstrung. Today, these therapies can cost as much as $1 million per patient, and they suffer from incredibly inefficient manufacturing, where it can take a very long time and a high degree of cost in order to get cells that are reliably able to take up genetic material. And that's frankly because getting into the nucleus itself is a tough affair. To date, we've been using good enough practices in order to get these therapies out there, which makes sense, but it's typically led to up to 50% of precious donor cells dying in the manufacturing process, and has been limited by the fact that you require cell division or mitosis in order to actually integrate into the genome, meaning it can take days to know whether or not you were even successful in your process. Also, there are physical limitations where your actual size of payload of genetic material is oftentimes limited to less than 5 kilobase pairs. However, critical safety genes, as well as elements of therapeutic interest, could be as much as 10 kilobase pairs, resulting in a mismatch of what's possible versus what we'd like to be able to do. As you can imagine, any kind of standardization of this process at any kind of a manufacturing level is a nightmare. Through the NSF I-Corps program, we spoke with over 15 stakeholders who were involved in this world of cell engineering and therapy. And what we heard time and time again are that modern techniques, specifically good manufacturing practice GMP grade techniques like CRISPR, lentivirus, etc., are expensive, they're complex, they require multiple days just to know whether you were successful or not, and most importantly, they result in preventable cell death. There's a large degree of room to improve both cost and viability in the process, and unlocking new abilities to put larger genes, their payload sizes, into the cell would change the entire landscape for how we think about viable therapeutics. We at FlowPoint seek to solve that problem using our Nest cell engineering platform. In a nutshell, our solution takes first principle approaches to think about a mechanical solution as opposed to a biological one. We employ microfabricated continuous flow channels where cells pass through small channels that have perforation needles that allow passive diffusion of genetic material directly into the cell and directly into the nucleus. There are several advantages here, with sub 30-minute integration time for DNA, where you can prove that your DNA is actually integrated well. More than 95% of cells survive this process with no issues, and it's fully agnostic to the actual payload in question and the technique in question of how you actually integrate into the genome. Notably, we're able to support much larger sizes of payload than any other technology out there and can process billions of cells per minute with no issues. Because this technique itself is very agnostic to the type of cell, you have additional applications in the world of synthetic biology and agricultural biology, where CAR T cell and cancer therapy is just the tip of the iceberg. We built this technology in partnership with Colorado University in Boulder, and thus far, data that's been published has been able to successfully show effective DNA integration and expression in less than an hour, proven restoration of the cell membranes, and standard growth curves compared to controls, proving that the cells not only survive, but they're also viable, less than 2.5% DNA damage, and most importantly, we've been able to show that from a cost perspective, we've been able to validate a silicon fabrication technique that has negligible cost of goods, especially compared to the more complicated biological methods that are used today. We plan to go after the actual markets in question here via a parallel path. First of which, a platform called Nest for Discovery, which is mainly targeting the public and private markets and sectors for people who are developing new therapies or are involved in gene editing at the lab level. In this process, we believe we can sell the device itself that controls pressure and the actual flow of cells for $20,000 as a one-time unit cost and a disposable consumable at $125 per unit. Meanwhile, on the industrial basis, as we're building a solid foundation of clinical evidence, our industrial target profile is that of CAR T cell and similar therapeutic manufacturers, industrial synthetic bio, etc. And we believe in this case we can emulate a lot of what companies like Maxcyte are doing, where we can have an embedded flow device for $200,000 in their manufacturing process, inclusive of licensing fees as well, and have a larger format parallelized nest silicon wafer consumable that we can demand $3,000 per run for. In order to be able to actually effectively get to a successful exit of this company, it's important to know where we are in the process. Thus far, we've been able to show some exciting initial feasibility data, but we're currently seeking a $50,000 research grant to fund additional performance data to show performance at varying cell types, various payloads, etc., to be able to build the foundation to start properly marketing to both industrial customers as well as other research labs for discovery purposes as well. We hope to be able to collect this data and publish by the end of 2026 and raise an additional Series seed of about $2 million, inclusive, as well as an STTR grant that will fund the commercial launch of the discovery platform and a lot of the development of the industrial platform. We hope to be able to secure key commercial partnerships and then raise a Series A that will allow us to scale rapidly to an exit event sometime in 2028 with an acquirer such as Thermo Fisher Scientific, Danaher, or others that are involved in the cell engineering space right now. When you compare us to the competition, obviously status quo we've already talked about, right? But even upstarts that are trying to solve the problem, like Portal or Basilard, the thing that's very clear here is that in terms of all of the axes of importance, can you get directly into the nucleus with large payloads? Can you express quickly? Can you have fast cell throughput? Are you a low-cost device? And do your cells survive? There is no one else who comes close to being able to meet all of these check marks like we are. In that way, FlowPoint's Nest device is an N of 1 in this market space, and we are poised to win on that basis alone. In terms of the team composition, we're an experienced team that has what it takes to get this thing to a successful exit point. I'm serving right now as the CEO or business lead, where previous to this, I ran a medical technology company called Geneticist, where we raised over $40 million all the way through to an acquisition by a Google spinout. I'm joined by a co-founder and someone who also was involved at a company called Proscia, who did cell pathology, who is serving as a commercial lead. His name is Piyush, and he's been able to prove over $50 million in both venture capital and strategic partnership success in the past. We're also joined by Sean Ding, who is serving as an advisor, scientific advisor, and is employed by Colorado University. He's a seasoned technology inventor, a microfluidic researcher, and has had 2 past cell manipulation ventures that he will be hopefully adding another one to here with FlowPoint. Overall, we feel well poised to tackle a very large $27 billion total addressable market, but also fairly large, sizable addressable, and SOM as well, where we believe that the cell engineering labs, manufacturers that are able to get us $150K average revenue per site with a pretty significant amount of ARR are a solid target. We believe we'll be able to have a penetration of almost $30 million within 4 years in terms of overall revenue, and a lot of that is based on the fact that we believe that we'll be able to secure key therapeutic partnerships by the year 2028 and really inflect at that point in time to be able to see very consistent and steady and healthy growth that make this to be a very compelling story overall. As part of the Embark program, our ask and on a priority basis is number 1, a $50,000 research grant to be able to support Dr. Ding's research and get us to a point where we have a foundation to be able to launch the product in earnest. And then as a lower priority, also a monthly stipend so we can have a laser-focused team that is focused on this project only. Ultimately, we believe it's unacceptable that cell therapy is as expensive as it is, and a lot of it is because you're looking at manufacturing processes and practices that would be unacceptable in any other industry. We believe with FlowPoint's Nest device, we can help tackle any cell, any payload, anytime. And it's in this way that we can make sure that cell therapy is accessible for all. Thank you.